As a high-level introduction, Juventas was spun out of the Cleveland Clinic almost 10 years ago. We are focusing on non-viral gene therapy strategies to activate endogenous stem cell-based repair of the body. We're focused on several large unmet clinical needs, a primary focus on cardiovascular, but today I will actually talk about some of the other indications that we are pursuing. And we also have a near-term inflection point with Phase 2b data coming out later this year. So I'll look forward to sharing that with you. Our lead molecule encodes stromal cell-derived factor 1, SDF1, which has now been shown by many labs to be a key regulator in repair of damaged tissue. What we know about SDF1 is that following injury, it has the ability to stimulate repair through multiple mechanisms. It recruits endogenous stem cells to the damaged area, it promotes new blood vessel growth, and it inhibits cell death, together leading to revascularized tissue that has less fibrosis. What we were also able to show in the early years was that to induce this repair, you needed approximately 2 to 3 weeks of expression, localized to the area of injury. So the strategy that Juventas took early on was to use non-viral plasmids to express SDF1 local to the injury. So our vector JVS100 is on the right, your left, and you'll see it's driven by a CMV promoter, CMV enhancer, and has an RU5 element in it. We have used this vector safely now in over 250 patients. It gives us the transient gene expression of about 3 weeks that we're looking for, regardless of tissue type to which we deliver. Importantly, it's non-immunogenic, highly scalable, and the cost of goods are nominal. Shelf lives have been demonstrated to be years at refrigeration. We have introduced JVS100 to 3 clinical indications, late-stage peripheral artery disease, and that's the primary indication I'll be talking about. We recently completed enrollment of a Phase 2b clinical study with data pending later this year. We have some very exciting Phase 2a data in heart failure. I've presented this before and won't talk about that today, but I would like to touch upon some of our dermal scar clinical data, as well as some new data we've been generating in the incontinence and muscle regeneration space. So our primary focus has been patients with late-stage peripheral artery disease, also known as critical limb ischemia. There's an estimated 2 to 3 million patients in the United States suffering with late-stage PAD for all the reasons that cardiovascular disease is growing: hypertension, diabetes, obesity. It's been primarily a device-managed field to date: vascular surgery, endovascular procedures such as stenting, grafting, and atherectomy. And the complications tend to be long-term amputations, non-healing wounds, difficulty walking, and significant pain. We had performed some time ago a Phase 2a clinical study in patients that were considered poor revascularization candidates with critical limb ischemia. It was primarily a safety study in 48 patients. We looked at 4 escalating dose regimens, or really 3 escalating doses delivered in 4 different distribution patterns. And what we're able to see in that population was, first, we hit safety, and we really have had a great safety signal really in all our clinical trials that we've run. After a year following a single administration of drug, we saw nice trends towards improvement in wound closure. We saw reductions in pain, improvements in Rutherford score, and improved quality of life. Interestingly, we had very few amputations in the full population. I think there was 4 across the full study, which is a trend that others have also observed. So while we're excited about that data, we struggled a bit with how to engage their target patient population. I think several people have seen that there's no option of poor revascularization CLI patient population is a challenging one, and particularly with amputation rates having been on the decrease due to aggressive device interventions, the studies become very large to run. So we sat down with our key advisors to understand where the population of patients is now and what the unmet need is. The field has really moved heavily towards below-the-knee revascularization, and that has been very successful in helping lower these major amputation rates by opening up macrovascular flow below the knee. The challenge, however, for these patients is they continue to lack blood flow in the foot, oftentimes due to other concomitant issues such as diabetes, end-stage renal disease. And what this leaves distal to where the blood flow ends at the ankle is non-healing wounds, significant microvascular disease, and a lot of pain. So this is an example of the type of patient we're engaging in our phase 2b clinical study, Engaged. And this is actually a patient who came into the Cleveland Clinic and is a standard story that they're seeing now. Patient comes in with a small wound in their toe. They do an angiogram, and what they see is a significant lack of blood flow in the lower limb. Now they can successfully go in, open the major vessels up with a stent, restore blood flow, and fortunately for this patient, they were able to avoid a major amputation that might have occurred maybe a decade ago. The challenge for this patient is that their toe wound continued to worsen. And that was due to the lack, while they opened up, as I like to say, they opened up a highway, they were not able to create any side streets due to the microvascular dysfunction of the foot, and therefore we never saw a reperfusion in the foot. And these patients, while it's great that they're able to maintain their limb, they go on to have minor amputations, transmetatarsal amputations, suffer with significant pain and ambulatory challenges. And what we know from some of the literature out there is this can be measured or monitored through the measurement of toe brachial indexes. So what the field has been able to show is despite successful macrovascular procedures, the TBI, or toe brachial index, oftentimes doesn't improve. And in the absence of that, what we see is a lack of healing wounds. And so why is this important? I mean, you saved a limb. Is a wound really that big a deal? The answer is yes. What we know is that post-revascularization, there's still about a 50% rehospitalization rate in the following 6 months. Amazingly, about half of that is due to non-healing wounds. If you're able to heal that wound, you reduce hospitalizations by about 30%. And if you're not able to heal that wound, the likelihood of a major adverse event in the next year is almost 4 times. So this is where the field has moved. It's really moved away from the no-option patient. It's successfully been able to create macrovascular flow, but the focus really is on microvascular dysfunction that occurs distal to the procedure. So taking that into account, we designed STOP PAD, and we've now enrolled that study with 109 patients. It's a double-blinded, placebo-controlled study in patients with advanced PAD and non-healing wounds that have undergone a successful below-the-knee revascularization, and despite that, continue to have poor blood flow in their foot. They received either one of 2 treatment regimens or placebo, and importantly, we re-administer the drug in 3 months to assess whether a single administration or 2 administrations are more beneficial for these patients. The primary endpoint is a composite that will look at wound closure, amputation, and worsening of the wound. I can tell you we have gotten guidance from the FDA that a wound closure endpoint will be acceptable for registration, so that's a big milestone for the company as well. The physicians and patients have both appreciated this study because what it is a simple office visit and injection of the drug into the foot anytime within a couple of weeks after the revascularization procedure. And, you know, for the proceduralists, the nice thing is we're not competing with them. What we're doing is we're trying to make their outcomes better. They already know they can restore macrovascular flow. They already know they can do a good job at limb salvage in a lot of these patients. What they also know is that they have very poor tools to be able to restore blood flow in the foot. So working together, the goal is to be able to enhance that wound closure as well as prevent minor amputations. We completed enrollment of the study in July of this year. We anticipate 3-month data from the single dose in December, and then we'll have in Q1, Q2 of next year the 6-month data from 2 doses. I have mentioned that JVS-100 really has application of a broad range of indications. We have historically focused a lot on cardiovascular disease, but what I wanted to do is share a little bit of what we've done outside that space today. So one of the areas we looked at was dermal scar prevention. Clearly a significant cosmetic issue for a number of patients. Hypertrophic scarring and keloid can also interfere with function and movement. What we knew early on was that SDF1 plays a significant role in wound healing. It has the potential to demonstrate anti-fibrotic, anti-scar, and what we wanted to look at is what happens if you dermally deliver JVS-100 in a patient immediately following an incision. Actually, I'll say it. In this early one, we did it with pigs first, and we looked at red Duroc pigs. This is a model for hypertrophic scarring. Each animal received a full thickness incision that was sutured as if it was a surgical scar, and immediately post-treatment it either received dermal applications of JVS-100 or placebo. And here you see the results. Placebo, you get elevated raised hypertrophic scarring consistent in all areas. I think interestingly, 28 days after in the treated pigs, all you see are the tattoo marks of where the incisions initially were, really no scar present. For us, we like this data for a couple of reasons. I think it's the most clear demonstration that we have an active drug. It demonstrates our ability to work outside muscle and skin, another large tissue type for which there's significant opportunity. And prior to this data, our focus had primarily been in chronic disease. What we are able to show that if you're able to intervene immediately after injury, you can prevent a lot of the downstream fibrosis and scar. And we've seen that in other indications as well. When we look at three-dimensional imaging, we see there's a significant less scar volume, significant less height, and overall that their skin is healed more uniformly. We were able to translate that into a small phase 1b clinical study. It was primarily safety focused, but we did design it in a randomized placebo-controlled dose escalation fashion to get some preliminary efficacy data. The study was done in patients following an open heart procedure, so sternotomy, which has 100% probability of scar. And all we did was immediately after closing, the surgeon used this dermal delivery device to apply JVS-100 to placebo along the edges of the wound. We saw safety again. I think that's the most important takeaway from this study. Interestingly, despite small populations, what we did see was trends, dose-dependent trends towards improvement in both a patient-observed scar assessment scale and a physician-observed scar assessment scale. And I think importantly, we also saw accelerated wound closure with the treated wounds having much less wound area than the non-treated or placebo-treated wounds. Not only is this, I think, exciting for the cosmetic or dermal post-surgical wound repair market, it plays directly to what we're trying to do in the STOP Ph.D. study where we want to heal dermal wounds. Another area where we looked at, and so just to back up on that, we're excited about this data. The next place we'd go with this would likely be in a more cosmetic-oriented indication, such as abdominoplasty, or other areas where they have significant post-surgical scarring. There's also opportunities in scar revision for hypertrophic or keloid scarring. What plastic surgeons and physicians know is that if you have one hypertrophic or keloid scar, when you have a procedure, you have a high likelihood of having another. So there are populations that are at risk for this. This data is kind of exciting because it really shows the breadth of what we're able to do. We know that JVS-100 SDF1 works really well in muscle and skin. As I said, when we first ventured into this, we were born at the Cleveland Clinic, strong reputation, cardiovascular, that's where we focused. But through some collaborations with the Cleveland Clinic, Case Western Reserve University, we've now expanded to look at the potential for muscle regeneration, specifically using incontinence models. So I will share a little bit about the fecal incontinence data we have generated with Cleveland Clinic. It is a disease indication that is significantly underrepresented and for which there is a huge market need because there really are very few tools that work for these patients. What we were able to show early on was that following a muscle injury, you have a significant recruitment of muscle precursor cells to that area that helps regenerate the muscle. And this was done in animal studies. We then took that into an fecal incontinence animal model, and this is a pretty severe injury in which they excise about half of the sphincter muscle, leaving the animals incontinent. And they then inject the JVS-100 or placebo and follow those animals out to 8 weeks. What we're able to see in that, which was quite exciting, was that while the placebo continued to worsen in function, JVS-100, whether alone or combined with stem cells or in the context of a scaffold, provided significant improved function of that muscle, improved contractility, which translated to functional outcomes in these animals. And when you look at the histopathology, what you see in the treated animals relative to the injury alone is a lot more new muscle. So a real regeneration and revascularization of that tissue. I could tell you we have similar data for sake of time I won't share right now in stress urinary incontinence models, and we've now, through partnerships with University of Miami, extended into demonstrating in bronchopulmonary dysplasia that we can help prevent lung fibrosis. So it really is a powerful therapy. We're very fortunate to have phase 2b data coming out. We're looking forward to that, and that's been our initial focus. But I think downstream there's really a number of different areas in which we could impact clinical populations with JVS-100. So just as a quick summary, JVS-100 has been demonstrated safe across multiple clinical studies. We have phase 2b data coming from an advanced peripheral artery disease study that we've recently completed enrolling. It is a platform program, and we've demonstrated both clinical and preclinical therapeutic potential in multiple indications. And as a drug modality, it has several attractive features, not to mention the affordability of the goods, the ease of delivery, scalability, and the biology being translatable from one organ to another. So with that, I think I have exactly 20 seconds left, and I don't know if anyone can fit a question in there, but happy to take one if they do. Thank you very much. Appreciate it.